I think maybe it's 8:00 and a good time to start, if that's okay. I'm Chris Fanta. I'd like to welcome you to Asthma Grand Rounds this morning and to an exciting program on the impact of air pollution and asthma, presented by two outstanding experts in the field. And before I introduce them, I just thought we'd review our usual process for submitting questions. Please text the questions to me. Jot down this number if you would, 617-513-6043, and we will put the questions to the speakers at the end of the presentation. And for those who want CME credit, if you'd send me your name and degree to cfanta@partners.org, I'll forward it to Harvard Medical School for the evaluation and the CME certificate. And then I'm hoping absolutely no technical problems today, but if you did, just quick send an email to bwhwebcasttroubleshooting@partners.org, and they are very good and responsive. Great. So this morning we have two presenters. Mary Rice is going to present first. She is a member of the Pulmonary and Critical Care Medicine Division at the Beth Israel Deaconess Medical Center. And Diane Gold, a member of the Channing Division of Network Medicine at the Brigham and Women's Hospital and professor in the Department of Environmental Health at the T.H. Chan Harvard School of Public Health. So thank you both for participating or leading the discussion. I'm going to stop share, and Mary, you're on. Thank you.
Hey, thank you. Good morning, everyone. Thank you so much for joining us, and thank you to Dr. Fanta and others for inviting me to speak today. So first of all, we have no relevant financial interest to disclose. And our hope is that by the end of this talk, you can identify outdoor air pollutants and mixtures that are risk factors for asthma, both exacerbations of asthma for people who have the disease and incidence of new onset asthma. Dr. Gold will describe mechanisms of airway toxicity due to air pollutants and interactions with microbes. And then I'll end with a discussion about interventions, and I hope that you'll be able to identify air quality interventions that may benefit asthma. So I'm going to start by a super quick overview of air pollutants and sources. So at a very high level, we're talking about combustion, whether it's combustion of fuel that we use for transportation or energy, or burning of biomass from indoor burning, or the big one being wildland fires. And when combustion occurs, there's a plume of pollutants that's released. And I'm going to take you through some of the big ones that have been studied in the epidemiologic literature. So first, fine particulate matter. I'm sure you're all familiar with this term. And PM2.5 refers to the size of the particles, not what they consist of. So particulate matter with a diameter of 2.5 microns is small enough to deposit into alveolar spaces. And it includes combustion-related particles like metals and organic compounds. And it also includes biological matter that Dr. Gold will discuss as well. PM10 refers simply to the size range, so 10 microns in diameter, and those particles enter the respiratory system and typically deposit in the larger airways, includes things like dust and pollen and mold. There's been a lot of work over the last several decades estimating exposure to the general population, and there's different approaches that have been used. One of the approaches is to use satellite imagery to estimate the density of the aerosol optical depth of the column of air above a spot on the Earth to estimate what the particle density is. But then the models use other sources of information, like how is the land used? Are there highways nearby? Are there power plants nearby, to try to estimate what the PM2.5 concentration is on a given day. And then after those estimates are made, the estimates are compared to what's actually measured on the ground. That's called calibration. And through those iterative through that kind of iterative process, we can arrive at an estimate of what the pollution level is on any given day on the map. Another approach increasingly is to use machine learning methods. This is an example of work by Tian Di and Joel Schwartz estimating elemental carbon exposure. Elemental carbon is a traffic-related constituent of PM2.5. This is one of the newer models. Nitrogen dioxide is a gas, not a particle, and it's produced when fossil fuels are burned. So when we're outdoors, NO2 is a really good indicator of traffic, and that's why I'm showing here a map of Toronto, and you can see how the NO2 levels correlate really well with where the roads are at a very fine scale in the city. But NO2 is also released from indoor sources like gas stove use. And it's been associated with airway inflammation, oxidative stress, lower lung function, respiratory hospitalizations. But importantly, it's also a precursor to ozone, which is the last pollutant I want to introduce you to. It is not directly emitted by the combustion sources, but it's formed through secondary reactions in the atmosphere. So when nitrogen oxides and VOCs are released, they undergo chemical reactions that can form ozone, and the formation of ozone is enhanced in the presence of heat and sunlight, which I'll get to later. And ozone is it's a well-established respiratory irritant. It's a very powerful oxidant, and it's a trigger of asthma hospitalizations. So those are the pollutants, but in the real world, our patients and all of us are exposed to a whole mixture of stuff. And sometimes the best indicator of pollution exposure isn't necessarily the estimate of one given pollutant on the map, but it's how close you are to sources. So, for example, here in the Boston area, traffic is a major source of air pollution. And we tend to find the greatest health effects just by looking at how close people are to busy roads, because living close to a road means you're not only exposed to really high levels of particles, but also gases. And here you can see how that plume of gases and particle numbers, which is a measure of the really fine particles, how it shoots up close to the roads and then dissipates to background levels about 200 to 300 meters away. And this brings me to a really important point of environmental justice. I mean, who do you think is going to be living closest to these combustion sources, the highways? Now, this is a picture of Chelsea, where I have my pulmonary clinic, and it's surrounded by busy roads. It's also close to the airport. It's in one of the most polluted parts of the Boston metropolitan area, and it's also one of the poorest areas. And it's most of the people who live there are racial and ethnic minorities, and so this is this triple jeopardy where the people who tend to be most heavily exposed to the pollutants are also those same communities that suffer external factors, social vulnerability, racism being a major factor, and poverty, psychosocial stress, and in some cases, biological susceptibility, like very young age or very old age. And these three forces interact to result in extreme health disparities. And the COVID-19 pandemic has brought this to public attention, but this is not a new issue and has been, and this is one of the major reasons why environmental health is such a major cause. Environmental exposures is one of the major causes of health disparities that we see in our patients. Here is a story that was published that got a lot of attention in the global media in December of 2020. This little girl who lived in London was heavily exposed to pollution throughout her lifetime and died of an asthma attack. And her case has drawn a lot of attention because it's the first time the coroner actually listed air pollution as a cause of her death. And even though we know from the research that air pollution causes asthma attacks, it never ends up in the medical record. So let me take you through what we know about triggering asthma attacks. This literature is very mature. There have been many studies over the last several decades showing that PM 2.5 and ozone is a cause of asthma exacerbations. And the EPA includes these in their cost estimate, cost benefit analyses for air quality policies. There's evidence that asthmatic kids are more susceptible to these pollutants than adults, which are likely due to factors such as the higher ventilation rate per body weight of children, the fact that their airways are smaller, that there is a higher deposition of PM in the tracheobronchial region of children, and also they spend a lot of time playing and exercising outdoors, during which time they're exposed. Here's just an example of some of the evidence of ozone and PM 2.5 in asthma hospitalizations. As you can see here, this is work that was done in Atlanta, and you can see a linear relationship between ozone exposure and asthma hospitalizations for kids, and a similar kind of relationship for PM 2.5. And there doesn't seem to be a clear threshold. I put these little red arrows where the current EPA standards are, so you can see quite clearly that ozone is a risk factor for asthma hospitalizations, well within the typical exposure range that the population has, and well within EPA standards. And the same is true for PM 2.5. I want to mention wildfires because they are a major source of air pollution, and they clearly trigger asthma attacks. Just some examples here, all from California. And this image here in the upper left from a wildland fire in 2008 shows the PM 2.5 levels before, during, and after a wildland fire event. And you can see that the PM 2.5 levels are orders of magnitude higher during the wildland fire event. And respiratory hospitalizations, which you can see here in the middle, increase in association with PM 2.5 levels. And that slope is much steeper for asthma. And in this particular, actually it was from a different study from 2003 Southern California wildfires. During the really heavy smoke conditions, asthma admissions went up by 34%. So really significant increases. And there's some evidence in the Medicare population that PM 2.5 from wildland fires may be more toxic for asthmatics than PM from other sources. So in that study by DeFlorio Baker, it was a 6.7% increase in asthma hospitalizations for wildland fire PM versus 1.3% increase for the same increment in exposure in non-wildfire PM. And some of that may be due to the fact that it's not just PM, right? When there's a wildland fire, there's a whole mixture of toxins that are released. Here's some innovative work that was done in Canada looking at inhaler dispensation during wildland fire smoke events. And in association, and as PM 2.5 went up during wildland fire events, salbutamol dispensations also went up. So I was just talking about triggering asthma attacks, and that literature, that's been known for a really long time. And what this audience may think is well established, but as actually the consensus is really only just starting to emerge, is whether or not whether or not air pollution causes people to develop asthma, to develop the disease. Here's just one example of some work done in Europe merging some really large cohort studies. So 14,000 kids followed until their teens. And you can see here, it looks like NO2 and PM 2.5 overall are associated with a higher risk of asthma, but these confidence intervals cross the null. And part of the reason may be that to estimate the effect of lifelong exposure or long-term exposure to pollution on asthma risk really requires information about where people lived and estimates of exposure across the lifetime. So what I just showed you used the birth address, which is just kind of a proxy for long-term exposure. So when I was a fellow, I started working with Diane Gold and Project Viva. And a real strength of this cohort study is that recruitment began in 1999, which happens to be right around the time that these PM 2.5 exposure models became available, because the EPA was then measuring PM 2.5 all around the country. And so taking advantage of these models, which kicked in the 1990s, we could actually estimate what the lifetime exposure to air pollution was in these kids. So one of the studies that we did was to look at asthma risk, and we found that kids who lived really close to a major road, so less than 100 meters, had about 3 times the odds of asthma in mid-childhood compared to the kids who lived further away. These are generally children in the Boston metropolitan area. We also looked at long-term exposure using the air pollution models. And for that, we found an association with asthma or what you should really call it early asthma or reactive airways, wheeze, in the younger kids, the 3 to 5-year-old age group, with a stronger association in girls. So you can see that the effect estimates were larger in the girls for black carbon and PM 2.5. And we found the same kind of pattern for current asthma in mid-childhood. I'm just going to briefly touch on lung function. We did a lot of work looking at lung function, which is not the same thing as asthma, but I want to highlight here that we found a really consistent association between living closer to a major road as a proxy for that plume of pollution, and exposure to PM 2.5 and black carbon, which is a traffic-related component of PM 2.5, and lower lung function in these kids. And I want to bring it up because lung function in childhood is a predictor of lung function in adulthood. And so here we're showing an association across thousands of children and a shift downward of their lung function among those who are more heavily exposed to traffic-related pollution and particles. And if the whole population is shifted down, that means from a clinical standpoint, you're going to have a larger proportion of adults who meet the spirometric definition of COPD in adulthood. So I talked a lot about particles. When I was doing that work in Project Viva, we actually did not at that time have a long-term ozone model. We're working on those questions now, but I want to highlight this work that was done in California, looking at long-term exposure to ozone in different communities and risk of asthma in kids. And here the findings were a little bit more mixed, that there wasn't really a robust relationship for ozone and asthma overall, but there was among the kids who spend a lot of time outside exercising. And that's where people are exposed to ozone. So ozone actually kind of dissipates as it hits surfaces. So it's not really an indoor pollutant, it's an outdoor pollutant. I was fortunate to participate in this ATS workshop in 2019, where we were focused on reaching a consensus about that question: does long-term exposure to pollution cause asthma? We also looked at COPD. And we went through the epidemiologic literature, we went through the mechanistic literature, and we also had a clinical group that tried to determine if it was clinically plausible. And part of the backstory here is I mentioned that the EPA uses the association between pollution and asthma attacks in its cost-benefit analyses, but it doesn't incorporate asthma risk, which is a chronic disease. So the cost of more people in the US population developing asthma would be really significant and would really have an impact on the cost-benefit breakdown for air pollution interventions. So to just quickly summarize what this workshop concluded, looking at the whole picture, we felt like the evidence was compelling that traffic causes asthma in children. The story in adults was a little bit more complicated, and really more research is needed in this area. So now I'm gonna switch to Diane, who can talk about mechanism.
So if you could unshare, Mary, that would be great.
Okay, right.
Great. Thanks so much, Mary. I'd like to start by acknowledging, let's see. Can you all see? Can everybody see?
Yes, we can see.
Okay,
Yes,
great.
good, Diane.
I'd like to start by acknowledging some of the many colleagues whose work I'd like to share with you today, giving a special shout out to Dr. Stephanie Shore, Jinnah Park, Joanne Sardillo, and Andres Cardenas. Over many years, toxicologic studies and human exposure studies have provided evidence for biologic plausibility of associations of individual criteria pollutants and mostly short-term asthma health effects. And these studies have contributed to the EPA integrated science assessments and to pollution regulation. Non-T2 mechanisms predominate in short-term pollution effects. For example, these include pollution-related increases in oxidative stress, IL-6, C-reactive protein, and the endothelial and leukocyte-associated protein ICAM1. There's limited evidence, however, on how air pollution increases asthma incidence, on distinct mechanisms of action for specific particle pollution components, and on the combined effects of multiple pollutants and multiple co-exposures. In the real world, as Mary said, we do not encounter one exposure at a time. Epidemiologic studies enable evaluation of asthma risk in response to multiple pollutions, some potentially protective and some deleterious. For example, here in Project Viva, which Mary's mentioned, in children with elevated prenatal exposure to PM2.5, prenatal vitamin E showed a stronger and larger association with exhaled nitric oxide levels in adolescents. But prenatal fatty acids were found to possibly protect against prenatal PM2.5 effects on the risk of teen asthma. Outdoor and indoor particle pollution has many components that may act in different ways and vary spatially and temporally. Despite decades of research, there's still uncertainty as to which components are responsible for adverse effects of particle pollution on asthma. And EPA regulation is based on overall particle pollution level. Here you see in Boston how particle components measured by X-ray fluorescence cluster by the sources of the particles: biomass fuel burning, road dust, secondary pollution, including pollution from power plants, motor vehicle pollution, and marine aerosols. Recently, Dr. Petros Koutrakis and his colleagues have demonstrated the presence of spatially and temporally varying radiation on particle pollution and its association with systemic inflammation and reduced lung function. He's working with Dr. Pipitanical's group to assess particle radiation effects on the asthmatic children in her school-based cohort. Here you see the seasonal variability of particle radiation in various parts of New England. Endotoxin, or LPS, a bioactive component of gram-negative bacterial walls that's a toll-like receptor agonist, is also on particle pollution. This figure by Dr. Duyong Park, Don Milton, and colleagues demonstrates seasonal variability of outdoor particle-associated endotoxin. Our group and Dr. Peggy Lai's group have demonstrated effects of endotoxin on children with asthma. Fungal components that are also toll-like receptor agonists and produce proteases are measurable on a large range of air pollution particle sizes, including respirable particles and particles that enter the nose and upper airways. Note that while components and DNA for microbes is measurable on particle pollution, it's likely that a high proportion of the microbes detectable by sequencing are not viable. Nevertheless, the fungal particle components, including TLR agonists, proteases, and allergens, could still influence asthma development or asthma control. The effects of individual air pollution PM components or gases on the airway epithelium can be assessed in vitro or in mouse models. As many of you know, lung epithelial cells are not just a passive barrier. Air pollutants and their components in combination with other environmental exposures may disrupt the epithelial barrier and tight junctions, alter mucociliary function, activate innate immunity, promote T2 immunity and allergic asthma development, promote asthma development through non-T2 mechanisms, and make asthma harder to control. Here you see that air pollution and the enzymatically active pollutant components could destroy the tight junctions here and allow easier ingress of allergens and microbes, influencing both the integrity of the epithelium as well as immune development related to either atopic or non-atopic asthma mechanisms. The epithelium also has toll-like receptors that detect a range of bacterial, fungal, and viral components with downstream innate immune signaling with import for asthma. Here you see another figure depicting the respiratory epithelium with its ciliated, goblet, and basal cells and tight junctions, and aspects of normal respiratory epithelial function, including antimicrobial action and mucociliary clearance of microbes and debris. Surfactant on alveolar respiratory epithelial surfaces not only serves to keep open the alveoli, but it's also a barrier for nanoparticles. And air pollutants may influence asthma through interfering with normal lung handling of microbes, including respiratory viruses. Air pollution can be one of a number of environmental exposures that leads to a diseased epithelium. Here you see a cartoon depicting epithelial cell structure in normal respiratory epithelium compared to diseased epithelium, where the tight junctions are disrupted, the desmosomes and other cellular proteins linking the cells to each other are broken down, and as with COPD, goblet cells have proliferated. Now, the asthmatic epithelium, depending on the asthma endotype, may not have such overt transformation of the epithelial structure, but more subtle differences still may alter epithelial function. In Dr. Jinao Park's lab, Meg Parker, in collaboration with Joanne Sardillo, cultured human bronchial epithelial cells from adults with and without asthma at an air-liquid interface until the cells created an epithelial cell layer complete with ciliated goblet and basal cells and tight junctions. In the cultures from human cells with and without asthma, they then assessed epithelial cell gene expression via RNA-seq in cells stimulated by endotoxin alone, house dust mite alone, both exposures compared to controls. Gene expression in response to endotoxin differed in the bronchial epithelium of those with asthma compared to those without asthma. For example, LAMB2 was differentially expressed in the epithelial cells of those with asthma stimulated by endotoxin. Laminins are the major non-collagenous constituent of basement membranes that have been implicated in a wide variety of biologic processes, including cell adhesion, differentiation, migration, and signaling. Mary talked about ozone, demonstrating non-atopic mechanisms for asthma in their obese mouse model. In Dr. Shore's lab, Dr. Cho and colleagues showed that mice exposed to ozone without allergen exposure were found to develop epithelial cell responses and downstream airway hyperresponsiveness with neutrophilic inflammation mediated by IL-33 and type 2 innate lymphoid cells. Moreover, ozone effects on the airway epithelium and downstream airway hyperresponsiveness were diet, gut, microbiome, sex, and endocrine dependent. Dr. Jack Harkema also evaluated non-atopic mechanisms for chronic rhinitis and asthma development in his mouse model, where repeated ozone stimulation led to epithelial cell death, mucous cell metaplasia, and type 2 lymphoid activation. Air pollutants may damage the airway epithelial barrier with epigenetic modifications of barrier genes, also potentially shifting microbial community composition of the human host. We're partially testing this hypothesis through the Children's Birth Cohort Project Viva in Dr. Sardillo and Dr. Cardenas' study of air pollution and airway cell methylation in adolescents. Nasal swabs were taken from the anterior nares in an anatomic location where airway epithelial cells predominate. Exposure to air pollution in the past year, that's past year's PM 2.5, was associated with changes in nasal DNA methylation in genes for solute transport, innate immune responses, apoptosis, cell cycle division, DNA repair, as well as T cell activation. These changes were distinct from those for allergy, allergic asthma, and exhaled nitric oxide. Functional gene categories represented in exhaled nitric oxide, asthma, and allergy included eosinophilic activation and T2 responses, which is not surprising in a cohort where almost all the children with asthma by the teen years have allergy. Differentially methylated sites specific to exhaled NO, interestingly, included solute transport, intracellular membrane trafficking, oxidative stress, and mucin production. In a pilot study supported by the Harvard School of Public Health NIEHS Center, Dr. Sardillo has worked with colleagues from the Jackson Labs to characterize via 16S sequencing the nasal airway microbiota in the same children from Project Viva for whom nasal methylation was measured. They defined so far 6 unique microbial clusters in the airways of these young teens. The clusters divided into those dominated by Propionibacter, Neisseria, Staph, Strep, Corynebacteria, and Moraxella. And they found preliminarily that air pollution, that's PM 2.5 in the past month, was predicted which microbial cluster the child was in. So in summary, air pollutants and their components in combination with other environmental exposures may disrupt the epithelial barrier and tight junctions, alter mucociliary function, activate innate immunity, promote T2 immunity and asthma development, promote asthma development through non-T2 mechanisms, alter airway microbial colonization, and make asthma harder to control via T2 and non-T2 or T1 inflammatory mechanisms. At the allergy meetings a week ago, our colleague, Dr. Elliot Israel, presented a wonderful talk on personalized therapies for difficult to treat asthma. He pointed out that while asthma may be predominantly T2 or predominantly T1, the airway inflammation that accompanies asthma exacerbations often reflects a mix of the two processes occurring in the same person. As Mary has said, there's an environmental and social context within which asthma develops, and asthma control worsens in susceptible individuals. Perhaps at the same time as multi-pronged personalized approaches are developed for individuals with hard to treat asthma, neighborhood, community, region, and nationwide approaches to air pollution reduction would damp down asthma development and improve asthma control related to any combinations of the mechanisms that I've just described. Moreover, as local outdoor and indoor pollution exposures contribute to community-wide inequities in asthma, pollution control measures are likely to contribute to environmental justice. With that, I'd like to turn the podium back to Mary.
you, Diane. So I'm now going to switch gears and talk about interventions. And I'm going to start with that outer circle of policy, and I'm going to end with patient level interventions that we as clinicians can offer. So big picture policy. So air pollution can be addressed through air quality regulations, through energy policies, and through a variety of other intervention policy level measures that address environmental justice. This is just an example of a hearing in the Environment and Public Works Committee that I participated in through the American Thoracic Society. And the ATS is very active on environmental policy on all of these issues, and a great forum in which pulmonary professionals can engage on national policy issues. So the Clean Air Act of the EPA, which regulates the major air pollutants that we've discussed today, nitrogen dioxide, PM 2.5, ozone. Overall, it's a huge success story. So since the 1970s, we have seen a dramatic decline in the concentrations of these major pollutants, largely as a result of this national policy. And in fact, it's even been cited as the most beneficial public health intervention of all time in the United States. And because of that success, we can now look at what the health benefits are of those changes. And this landmark paper by Erica Garcia and JAMA in 2019 looked at the decline in pollutants and was able to estimate the median decrease in asthma incidence in these California communities, holding all else constant. Because of course, air pollution isn't the only cause of asthma, but this study design really enabled the investigators to estimate that the change in asthma incidence in association with the change in air pollution. But the picture is not all rosy. Climate change is really complicating many things, but one of them is air quality. And as I mentioned early on, ozone, which is that secondary pollutant formed through reactions, well, on hotter, sunnier days, ozone formation goes up. And it's been demonstrated that because of climate change alone, a lot of these measures to reduce the emissions of the precursors of ozone are actually undone by the climate change effects. And this example here is in California, estimating that by 2050, all the measures to reduce emissions by that time are being undone by the increase in ozone resulting from higher temperatures. So we can advocate at the national level around policies. We can also advocate at the local and state level. Alex Rabin is one of our pulmonary and critical care fellow graduates from the MGH BIDMC program. He's now in Michigan. And he invited me to come with him to this local hearing at the State House around clean energy policy for the state of Massachusetts. And as you can see, there's people there in the background sitting on the floor. It was a completely packed room, and lots of people came up and testified. And there was a lot of chatter, and it was kind of noisy. It was kind of hard to hear people testifying, but not when we spoke. People really were interested in what the doctors had to say about this issue. And Alex is not an air pollution expert, but he is an expert. He's a pulmonologist, and he was able to talk about what air pollution does to his patients with chronic lung disease and how they would benefit from policies that reduce exposure. Here's just in the bottom left-hand corner is a staffer briefing. Now, our legislative staff want to learn about these things, and as health experts, we can tell them about how air pollution is a trigger for asthma exacerbations and a cause of childhood asthma when it comes to traffic-related pollutants. And local interventions can really make a difference. Here's a really concrete example of a traffic reduction program that was done in the 1990s during the Olympics in Atlanta, and it really worked. Public transportation went way up by over 200%. but traffic counts went down by 23%. And in association with that, air pollution levels got better. Ozone levels went down by 30% during the several week time period of the 1996 Olympics. And in association with those changes, asthma events went down by on the order of 44% decline for Medicaid and Kaiser asthma claims for kids. And then after the traffic reduction program ended, asthma emissions went back up to the same rate that they were before the reduction, the intervention. And this wasn't the case for other kinds of emissions. So really, air pollution, this is just, I'd like to give this example because it's very concrete. You look at the asthma events before, the asthma events during, the asthma events after, and it just went down and right back up again. And there's other examples like this, including the Beijing Olympics. Many cities have started to adopt low emissions zones to restrict traffic in the city. London is a great example of this. And the ultra low emission zone in London has been found to reduce NO2 by 36% since 2019. And it's been estimated that that reduction, and if it keeps up, is going to save 1 million pollution-related hospital admissions by 2050, which is a really big number for a city, you know, because we're not talking about national level differences here, just in the city of London. I want to focus on school buses because because they tend to be really dirty. Diesel buses are among the most polluting. Traffic diesel particles have been found to be especially harmful to the airways. And the school bus commute is a major source of black carbon, which is an indicator, which is a component of diesel pollution. It's a major source of black carbon exposure for kids. About 30% of their total exposure in childhood is from the school bus commute. And there's lots that can be done to make school buses cleaner, with the best intervention being switching completely to electric buses. Sarah Adar did this interesting work looking at taking advantage of an intervention to clean up school buses and looked at asthma events and exhaled nitric oxide before and after the intervention in kids, and found that among persistent asthmatics, the school bus interventions were particularly effective in reducing exhaled nitric oxide and reducing school absenteeism. So these are city level kinds of interventions, right? We can't really, our patients can't implement these kinds of things. But what do we tell our patients? And what can they do to protect their health? And this is especially important for the issue of wildfires, because the pollution levels, as I showed during those graphs, are extremely high during wildfire events. And we really need to be able to give clinical advice. And I participated in this ATS workshop that we published last year that really called for the need to identify effective interventions for the public and for patients to try to reduce morbidity from wildland fire smoke. And I talked about the great success story of the Clean Air Act, and that is generally true for PM 2.5, but not everywhere in the country. So on the East Coast, where the map is purple, pollution levels have been declining since the 1980s as a result of the emissions reductions. In the Northwest, even though emissions have reduced, PM 2.5 levels have actually gotten worse during that time period, and that's a result of wildland fire smoke in the United States and traveling across the border from Canada. So what can people do? Well, I think the best evidence is for HEPA filters. Everyone knows what HEPA filters are now. They really work in reducing particle levels. There's no question that these absorbent filters reduce PM 2.5 concentrations by significant amounts. Our work in the Boston area has found on average about a 50% reduction in PM 2.5 concentrations with long-term HEPA filter use. And aeroallergens are also particles, microbes. They all reduce proportionally when people use HEPA filters. Now, keep in mind, they have to be maintained. You have to replace the filter and all of that. So that is one of the challenges. But some of the best evidence for health benefits is for childhood asthma. And there have been a couple of meta-analyses, all of which have been pretty small, but have generally concluded that there is an improvement in the asthma quality of life with HEPA filter use inside the home. And there's also some research showing benefits for wildland fire smoke. What about N95s? I think several years ago, pre-pandemic, people wouldn't even be asking this question. But now that we're all wearing masks, you may be wondering, do these help reduce our exposure to pollution? Well, surgical masks do not. The air just, it's going to follow the path of least resistance. And that will include all the pollutants in the ambient air. But N95s or respirators are actually filtering face pieces that do reduce pollution exposure. They don't reduce exposure to the gases, but they do reduce exposure to the particles. There's some evidence that N95 use can mitigate the short-term physiologic effects of PM 2.5, but that evidence is all limited to healthy adults. And there aren't any respirators actually technically approved for kids because NIOSH approves N95s. I spent the last year and a half on this National Academy of Science, Engineering, and Medicine committee focused on this exact issue. There seems to be this huge gap in guidance for the public on respirator use. And I, as a clinician, don't know what to tell my patients when they ask. The CDC has been sort of trying to keep up with the pandemic and giving advice for how the public might protect themselves from COVID. But really, this question is much bigger than COVID-19. There is a rising incidence of wildland fire smoke. There will be more pandemics in the future. And there clearly are situations where it makes sense for the public to wear respirators, such as acute wildland fire smoke exposure, where you just have to get from one place to another. So this committee called for a new entity within Health and Human Services that would interface with EPA and the CDC to actually identify hazards, estimate the risk, identify appropriate respirators, ensure there's adequate supply, and actually create guidance that's targeted to different groups of the public, including kids and people with chronic disease, including asthma. We also had recommendations for the occupational side because people are exposed to pollution and microbes at work, and it may not, and this includes jobs that aren't traditionally under OSHA's authority. So the key takeaways that I'd like you to take with you are that air pollution exposure at contemporary levels, our everyday exposure levels here in the Boston area, cause asthma attacks, and when it comes to traffic-related pollution exposure, increased child asthma incidence. Low-income and racial minority communities are often most exposed to combustion-related pollution, and wildland fire smoke pollution is a major asthma hazard that is getting worse. Interventions that can improve air quality and can mitigate asthma. This has been shown for national air quality and emission standards, local urban low traffic zones and cleaner vehicles like school buses, and air purifier use in the home. And we as physicians who understand what an asthma exacerbation means and how it could be avoided by reducing exposure to pollution, we have a special role to play here to address environmental justice by engaging at the national, local, and patient level to reduce these exposures. I'd like to acknowledge my collaborators at Beth Israel, at the American Thoracic Society, through which I got very engaged on policy advocacy, my collaborators at the Harvard School of Public Health and Brigham and Women's Hospital. And I'd like to thank my family, Tessa, James, and Adele, and my husband, Ed. Questions?
Thank you so much, Mary and Diane. Terrific presentation, compelling evidence, and call to action, I think. And we do welcome your questions. Just send them to, remember, 617-513-0220. 6043. In the meantime, I thought I'd ask about the PM 2.5. And you had mentioned, I think, Mary, a little bit about differences perhaps in the setting of wildfires versus other forms of combustion. It feels like a bit of a black box. What do we know about those particles? And do you think of them as inert irritants or chemically active particles, or any details about that big category of pollutants?
Sure. So the best evidence of mechanism for how these broad categories of pollutants trigger adverse health effects, not just asthma, but cardiovascular health effects, is through oxidative stress. So the smaller the particles, the greater the oxidizing potential is. So tiny metals have been shown to trigger oxidative stress in the airway epithelium. Ozone, being a powerful oxidant, also falls in that category. I mean, other mechanisms that are involved are actually the transportation of the tiniest particles across the airway epithelium into the bloodstream. It's not clear how relevant that is for asthma in particular, but there is evidence that the tiniest particles actually get across into the bloodstream through isotopic labeling. But it's a whole gamish. And so that's actually something that we as environmental health scientists discuss at length, because PM 2.5 is just an indicator, right? It's just particles and how big they are. It doesn't tell you what it is. And it's pretty clear that the toxicity does vary based on the composition. But when you try to do the research to figure out which component of PM 2.5 is the worst, it can be really hard to disentangle because the sources tend to be the same. So if someone's more highly exposed to carbonaceous particles, they're probably also going to be more heavily exposed to the metallic components. And so really trying to tease all that out can be really tricky. There is a call for refining the way EPA monitors and regulates pollutants, because to try to get at the specific components that, for example, just to give you one, is black carbon. Like maybe we should actually be measuring and regulating that instead of PM 2.5, which includes all the potentially inert dusts and so forth. So
I have a question from Susan Corrick, who you know, and she was hoping that you would expand on specifics about HEPA filter devices. You know, there's so many on the market and some are variable in their effectiveness. Do you have any specific guidance?
Oh, sure. I can say a little bit on that. For full disclosure, my R1 is a HEPA filter trial in COPD. So I spent some time looking this up. The key thing I would say is you want to make sure it says HEPA on it, right? There are air cleaners that are not HEPA. And so that is an important thing to pay attention to as a customer when you're advising your patients. The other thing that I would say is some of them are oxidizers and actually generate ozone pollution. So I would advise against getting any HEPA filter that says it also is an oxidizer. Then I would turn to just the room size. So when you look on the box, so if it's powerful enough for a bigger room, I think at a very simple, just to keep it kind of simple, that's going to be a larger number of air changes per hour. So if you want a super powerful one, you could potentially advise getting one that's sized for an even bigger room, but at least get one that's sized for the room that you're going to use it for. Otherwise, it's not going to be powerful enough.
Diane, I had a question for you. Go ahead.
Sorry. You might want to ask around about the amount of noise that it makes at a high flow, because if you put a HEPA filter in a room and it's very noisy, people tend to turn it off, which is not what you want.
Diane, my question had to do, I think, with the Viva study. You know, at the Channing Lab, as you know, there's so much interest in prenatal exposure and the development of asthma. Were you able to sort out any information about maternal exposure to pollutants and the incidence of asthma in the offspring of highly exposed mothers?
Yes, to some extent, prenatal PM 2.5, distinct from exposures after birth, was associated with increased exhaled nitric oxide levels, measure of inflammation by the time the children were teenagers. But I think, Mary, you found that when it comes to lung function, at least, the exposures, the lifetime exposures and the early life exposures after birth had stronger associations with lower lung function. Is that right?
Mm-hmm. Yeah. So we did look at prenatal exposures, and we did have exposure in early life and exposure during pregnancy tended to be really highly correlated. So that made it a little bit difficult to tease out, starting with the asthma piece, whether it's prenatal, early life, or lifetime. But we did find associations with asthma risk for all of those exposure estimates. For lung function, we did find, generally speaking, that the more recent exposures had the strongest effects on lung function. We have another paper that's in press showing that as air quality improves, lung function growth was faster. So some of these effects on lung function, the good news is they may be reversible if we can improve air quality.
There was a question Nora Barrett had asked about potential value of the PM 2.5 detectors, which are it can be individualized. And do you see a role someday for people monitoring air quality and adjusting their activities according to the exposure that they as an individual are experiencing based on their pollutant detector?
Should I take that one, Diane? Okay, so I did a whole study having patients carry personal air monitors with them, and I also participated on a workshop, another ATS workshop on this very issue, patient level monitoring, something like that was the title. And I have thoughts on the current situation and the future. So current situation, there's a ton of market products out there. They're not regulated, and a lot of them are garbage. So I certainly would not recommend telling your patients to go out and buy air monitors and drive themselves crazy trying to figure out, what's an acceptable exposure, because there's so much noise there. However, even with current technology, I think there's a real role for communities to be able to monitor their exposure and identify hotspots. So the EPA doesn't have a monitor everywhere. And in fact, a lot of them are deliberately placed to get background pollution levels, not plumes, not like those clusters in Chelsea that I was talking about, where there's multiple sources. So it's a way for communities to be empowered and to identify that pollution is a problem. We can direct our patients to simply the EPA Air Quality Index, which tells them what the outdoor air quality is to regulate their behaviors.
And I want to expand a little on what Mary just said. I think it's important to empower communities to monitor their own neighborhood pollution, and to not put the whole onus on the individual, who often as an individual has less control over their lives than the community might have in controlling exposures.
Great. Well, the hour is up, and I just want to thank you for the terrific, stimulating, provocative, all around interesting talk. Thank you so much, Mary Rice and Diane Gold, and wish you all a happy day.
Thank you so much.
Bye.
Bye-bye.